12 o'clock on the dot here in the Pacific time zone. So we will go ahead and get started. So just a reminder that we do have some community guidelines for this conference. So if you could take a second just to read these on your own and make sure to honor them as we present and move into the question and answer space. I think that OSA has done a really nice job of cultivating an excellent conference, and I think reminding us of all these things is really important. So I think with that, I'm going to share my screen first here, and then I'll give some introductions. So okay, so my name is Brigid Meints, and I am an assistant professor, senior research at Oregon State University. I'm presenting in this session, and I'm also moderating in this session. And so just a few reminders as we get started. This is a recorded session, but we'll only capture active speakers. So if you would prefer not to be included in the recording, you can use the chat to participate. A video replay will be available in the Commons. I'm imagining that a lot of you probably have spent quite a bit of time on Zoom in the last couple of years, but in case you haven't, we do have this chat function. You can see that Michael, who is our tech manager for this session, has already posted something in the chat asking members of the media to get participants' permission before quoting words and content. I think for this presentation, we'll see how many people show up, but when we get to the Q&A session, we will most likely just be taking questions by chat. At that point, if there are a manageable number of people and people would prefer to unmute themselves and ask questions that way, we can also do that. In this session, we do have 4 speakers, and then we'll open up for Q&A. And then again, if you need any general support during the conference, we ask that you email the help desk at seedalliance.org. And then if any conference culture issues arise, we would ask that you get in touch with Kit Healy at kit@seedalliance.org. So I think that covers sort of the general overview of the session. I'm going to go ahead and introduce the speakers. So in addition to myself, we have 3 graduate students, Karl Kunze from Cornell University, who's a PhD student working with Mark Sorrells, Chris Massman, also a PhD student at University of Wisconsin-Madison, working with Dr. Lucia Gutierrez, and Cristiana Vallejos, who's a master's student here at OSU, also working with me. And so all of us are involved in this project, this national project together, and we'll be presenting on different components of the project. And so I'm going to start with a general overview of the project, as well as digging into one of the diseases that we work on. Karl is going to talk about weed competitive ability. Chris is going to talk about threshability in naked barley, and Cristiana will finish it up by talking about resistance to embryo damage in naked barley. And then we'll have time for Q&A. So thank you all for being here, and I am going to get started. Just to confirm, everyone is seeing the appropriate screen. Correct? Yes, great. Okay, so Again, we're here to talk about breeding or developing multi-use naked barley for organic systems. So, nope, doesn't want to advance. There we go. Okay. So just a quick overview of organic barley production in the United States. So as I imagine we all know, but perhaps not at this conference, given that we're all interested in organic seed systems, the demand for organic products is growing rapidly. Barley is the second most widely grown organic small grain in the United States following wheat. However, while there are about 2 million acres of barley grown total in the US, only a very small portion of that is grown under certified organic production. So according to the National Ag Statistics Service, as of 2016, only 51,000 harvested acres of certified barley were harvested in 2016, worth about $17 million. And there are a number of reasons for this. Most barley in the United States is grown for malting, for brewing and distilling. And there isn't as much demand in the organic beer market as there are in perhaps other food markets. And additionally, most of the varieties that are being grown have been bred under conventional systems. And so, as is something that we talk a lot about in organic seed systems, selections that are made under organic conditions during the breeding process are often better suited for organic production once they've reached released variety. And so with this process in mind, breeding is at this point for barley about an 8 to 15 year process, depending on what breeding techniques you use. And so it was important to us before we sort of started this breeding process to identify our germplasm resources and figure out what traits were of interest to us, as well as what germplasm might be useful for the breeding process to breed specifically for organic varieties of naked barley. And we did identify pretty early on that disease resistance and weed competition were going to be some of our most important traits to focus on. So just to contextualize what we're talking about here, what is naked barley? Some of you may be asking yourself, is that? So here on this slide, we have covered barley at the top, naked barley at the bottom. Most barley grown in the US and around the world is covered barley. That hull is essentially glued to the outside of the kernel, and so is quite difficult to remove, but can be done using mechanical abrasion. And so we can remove that usually through a process called pearling, which removes some of the bran and germ as well as that hull, and that's typically done for a food end use. Naked barley, shown here, is a whole grain kernel that can be eaten as is, well, post-cooking or milling, and doesn't have that unpalatable hull in there. And so for food end use, naked barley is typically of interest. That difference is caused by a single gene that is a result of a natural mutation that occurred about 8,000 years ago. And as I've already described, the end uses of naked barley are stratified a bit by whether or not they have a hull on them. So the three main end uses of barley are feed, food, and malt. And so around the world, animal feed is the greatest use of barley. In the United States, as I mentioned already, it's malting and brewing. Food end uses are only about 4 to 6%, both in the US and globally. And so sort of our goal as breeders with this multi-use naked barley project that we're working on is to develop a variety of naked barley that could work for all 3 end uses. So at this point, growers, if they're growing a covered barley, for example, and it doesn't meet malt spec, which is quite a difficult thing to do anyways, but with climate change is becoming increasingly difficult, they have to sell into the feed market at quite a loss. And additionally, if you're growing barley for food and it doesn't meet spec, you also have to sell into the feed market. And what we would love would be for growers to have the option to sell first into the malt market, if that's what they're going for, then into the food, and then into feed, because the food market has lower specifications required for quality than the malting industry does. So that's sort of to set the stage of what we're doing here. Naked, multi-use barley for organic systems. And so this project is funded by the USDA NIFA. Economic surveys to look at organic barley production currently in the US. And this was conducted by Brian Baker, who is a consultant on the first part of the grant. And so we put out a survey to organic barley producers around the US to look at current production, challenges that they may be facing that we as breeders could address, and their interest in growing multi-use naked barley. Additionally, then Brian conducted 19 follow-up interviews where we were able to take a deeper dive into their production. So just to sort of present some brief results from that survey, we did ask growers about disease issues that they face, and we were quite surprised to find out that 45% of them said none. Given what we have seen ourselves in our trialing and just sort of talking with growers. But that being said, we did have a few responses where, you know, growers were like, well, I'm not a plant pathologist, I don't know. And so it's possible that this doesn't, you know, capture the full picture. But some of the diseases that we are working on, Fusarium head blight, smut, some of the rests were things that we're already working on. So that's great that those interests align with our breeding already. And then we were interested to look at traits that were ranked by importance. So yield was the #1 trait that people were interested in, and then the rest varied a bit with region. So if you're interested in seeing the full results, I would recommend that you check out these publications. But competitiveness with weeds and disease resistance, as well as sort of more regionally important traits, are things that we're working on in this grant. So that was great. So I want to walk you through the germplasm that's being used for this project, and this keep these in mind because this is the same germplasm that all of the other speakers are working with. So we have sort of two main categories here. These are our regional elite trials. Barley can be both fall planted and spring planted. Typically we refer to that as winter and spring barley. And so we have 2 trials annually that each of the locations plants with sort of our most advanced lines, and then we can do more in-depth and use quality testing on these because they're larger plots. And then we also have our naked barley diversity panel. And so this is a panel of 384 lines, 254 of those can be spring planted. And so we have what we call the full panel as well as the spring panel. These include lines that we collected from breeder collaborators all over the world, as well as got resources, germplasm resources from GRIN and other germplasm banks around the world as well. And so they're all naked barleys, and there's quite a bit of diversity, as you can see in this picture here. And so then this panel, we're able to conduct genome-wide association studies on and potentially identify novel genes associated with the traits that we're looking at. So that's one of the benefits of having such a large panel, as well as this panel has been really useful in identifying parental material for future breeding. So a little bit more about this. So this panel has been genotyped with what we call the Illumina 50K SNP chip. So that is 50,000 single nucleotide polymorphisms. I'm not going to give you a genetics lesson here, but essentially it's a way to fully cover the barley genome so that we can associate genes or quantitative trait loci with the traits that we're seeing in the field. We did some kinship analysis, and we're able to cluster these into 6 subgroups based on a number of categories. So by sort of origin of breeding material, row type. So in barley, we can have 2 and 6 row head types. That's in reference to the number of seeds going up the spike on the head, and then whether it requires vernalization or not. And so that again refers to planting date. So winter types that are planted in the fall do require vernalization, spring types do not. And so these groups are fairly distinctive. You can see that a couple of them here trace back to a lot of the material that we got from GRIN, and then some of the other groups trace back to Pacific Northwest or more material that came from other parts of the US. And then these Pca plots here, again, perhaps offer a bit of explanation as to how this panel breaks up. So this is based on origin. So you can see we have, I think these are all Ethiopian lines here clustering together. These are all from Washington State. So this shows genetic relatedness essentially here. And then this one is based on vernalization requirements. So the ones that do not require vernalization or spring types are in red, and the ones that are winters or do require vernalization are in teal. So you can see that there is some clustering here based on vernalization requirement, but it's not as obvious as some of these based on origin. So now I want to transition into one of the projects that I particularly have been focused on, using all of the germplasm resources, as well as identifying this as a priority for organic systems. So smut is we actually have 2 types here that we focus on, both loose smut and covered smut. These are fungal pathogens that are seed-borne, and Covered smut is what we call an externally seed-borne disease. So the spores are typically, we call these smut balls. So rather than a normal head of barley, we have a smut head that's formed, and then it breaks up into these smut balls. These smut balls are sort of distributed through harvesting and cleaning, and the spores come off land on the outside of a barley kernel. And then when that barley kernel is planted the next year, that spore, this is oats, barley, and wheat, but that spore grows into the growing plant tissue and infects the head. And so we see this sort of cyclical pattern here. Covered smut can reduce quality because those smut balls are found in with the harvested grain. As you might expect, if you have a smutted head instead of a healthy head, for every head that is, you're reducing yield by that amount. So we can reduce yield as well with both of these types. Loose smut is what we call internally seed-borne. So on a loose smut head, the film on the outside of our little spore sac bursts, and the spores come out and are wind distributed, and they actually infect the flower of the healthy plant and infect the embryo. So that is then inside the seed rather than outside the seed, as with covered smut. In this case, because we don't have spores in the form of smut balls or on the outside, we don't reduce quality, but again, we do reduce yield. And why are these so problematic for organic systems? So in, I believe, the 50s and 60s, fungicidal seed treatments were developed that were so effective that they essentially reduced presence of this disease. And therefore, breeders stopped selecting for genetic resistance. And so most of the modern varieties do not have any sort of genetic resistance, or maybe are resistant to one race but not another. So resistance is race dependent, so we can have different races of the pathogen. And then there has not been really an effective seed treatment found for organic systems. So there are some hot water and steam treatments. However, these have to be done very carefully to avoid killing the embryo as well as the spores, and they're not very practical for large lots of seed. One of the only ways that growers can be assured that they don't have smut in their seed is to purchase double certified. So that would be a certified class of seed as well as organically certified seed. And there is very little production of double certified grain seed in the US right now, unfortunately. And so you may be forced to plant a variety that you're not interested in because it is one of the only types that has gone through this double certification process. And yes, and the other issue is that the resistance or susceptibility levels in a lot of varieties are unknown because they're not being tested for that. And so that is what we decided to do. So with the diversity panel, we inoculated all 384 naked barleys using this experimental procedure in the greenhouse. And so we collected those smut balls from field, and then have been assessing the plants as they've been growing out in the greenhouse. And so these are some preliminary results. The study is still ongoing. So we did a primary grow out. We're in the middle of the secondary grow out with all lines that showed resistance, and I should say potential resistance, because there is the potential for escapes that weren't properly inoculated. And then we'll do a third grow out. And the lines that show resistance through all 3 grow outs, we will still probably just label them as potentially resistant. But you can see, so this is broken out by spring and winter here. But we do currently, with the results of the second grow out that we have, 53% of the germplasm shows some degree of susceptibility. And so you can see that so far, and I should say so far, we're in the middle of our second spring grow out. So we haven't started the second winter grow out. So these results are only based on the first one. But we do see some interesting differences between the spring and winter genotypes, which is likely not due to the fact that they're spring or winters, but due to their breeding origins, and that likely we haven't been able to capture the full picture due to only having one grow out so far. This graph is to break the resistance and susceptibility levels out by the genetic subgroups that we see in the diversity panel. So if you remember back to the early slide, we have 6 groups. And we can see that there are some pretty major differences between the groups. And so quite interestingly, these 4 groups where we see high levels of susceptibility are all the modern breeding lines. And the 2 groups where we see perhaps high levels of potentially resistant lines are lines that we got from GRIN. So these would be, land races from areas around the world where barley has likely remained a staple crop, and growers may be growing under with fewer inputs, and so it's likely that they've been selected for smut resistance. So our job as breeders is to start working to introduce some of that resistance into our breeding programs. And then this is just, these are the same groups with the whole panel, and we do see one interesting difference. This group 2 here was for just the springs, the susceptible lines. There were many more of them than the resistance, and that has switched when we've added the winter lines in here. So that's probably where we were seeing a lot of that difference in the spring versus winter. So that'll be interesting to see how that changes as we complete this project. And then just really quickly, we did conduct a preliminary genome-wide association study to look and see if we had any places on the genome that were associated with this trait, and we do see a pretty clear peak here. This is based only on that first grow out. So we'll be updating this as we get more results. Based on the literature, there isn't a known resistance gene on chromosome 3H. However, there are a number of resistance genes that have been identified but not mapped. So it could be one of those, but it is not one of the major resistance genes that has been identified. And that could be because a lot of the work that has been done in Western Canada, where they likely have different races of smut than we do in the Pacific Northwest. And then just really quickly, we're going to be working on loose smut next. So we've done our inoculations already in the field. And so because this is internally seed-borne, we actually had to inject this sort of smut water mixture into the growing flowers. And we haven't planted this set out in the greenhouse yet. So stay tuned for those results in the future. And then this is sort of the overall next steps for this project is we're creating this very large population. In all 5 programs are working on this together. And so unfortunately, a lot of these lines that we've chosen turned out to be susceptible to covered smut, at least based on our greenhouse grow out. So more breeding work is still to be done, but it looks hopeful in that we will identify some potentially resistant varieties. And so then I just want to pull up my acknowledgements. So this is for the entire project. So these are all the collaborators that have helped me in the work that I've been doing, as well as the other folks. So I know I've gone on too long, but I think I'm going to stop sharing and then let Karl go next.
Can everyone hear me? Great. All right, I will share my screen here. Great. Which one shows up? Is it the presenter mode or the? Should be good. All right, so I'm going to be talking a little bit about components of wheat competitivity. It's been some work I've been doing, particularly with the spring barleys out here at Cornell. So Brigid covered most of this already, so I'll be quick in this part. This is part of the multi-use naked barley project. Trying the end goal is to breed organic naked barley for new environments. And we have various aspects of field research that we're working on. And yes, the ultimate goal for this project is eventually to develop multi-use naked barleys for specific regions. But I'll be focusing more on work on our spring variety trials. So there's a lot of challenges of growing organic naked barley that was already previously mentioned. Because it's organic, we have limited pesticide and herbicide options that we can use, or sometimes none at all. Cultural practice can account for some of the solution and perhaps a large solution. So what I mean by that is having the right crop rotations, breaking up disease cycles. And of course, that's kind of some research that is outside the scope of plant breeding. So hopefully with this work from the plant breeding side, we can work on making things a bit easier for organic naked barley to grow. And of course, because we don't have as many tools as we might in conventional sometimes, we have to have a greater reliance on the genetics of the plant itself. And of course, with weeds, having barley that's vigorous and as a result, that downstream wheat competitivity. So what is wheat competitivity? Wheat competitivity, as I'll define it, is the ability of a plant to outcompete or tolerate wheat pressure. Sorry, Mike. 1 second. There we go. So the first big point is weeds are really difficult to measure in a repeated experiment. There's multiple options we can do, and that's what we've been working on with this project for a number of years. And first is to measure the weeds where they are in the plot. And that's currently the practice for most of our variety trials. However, this can be very difficult as weed pressure can be inconsistent both across locations, certainly, but across years and within a specific field. Weed seed banks are not distributed evenly, and that's hard to do. So when you score this, in addition to looking at the weed pressure, it can be a bit subjective at times, and the weed pressure can be inconsistent. One solution that's been proposed in previous literature and other organic studies is to perform weed seeding, which in certain situations is something you should do or it would be a good idea. However, when we are planting at organic research farms, the organic research farms aren't too thrilled with the prospect of adding more seeds to the seeds, more weed seeds into the system. So that's something that we're limited in what we could do. So one alternative method we came up with is perhaps measuring something that's a little more reliable in the weed competitivity of the barley, and that's specifically the barley vigor. So how do we measure barley vigor? Conventionally, we've been doing this by visually scoring the barley throughout the vegetative growth stage. Traits that were most moderately heritable that we measured were vigor and early height. However, with this 1 to 9 scale that we visually score with barley, that lacks some precision. And this ordinal scale can be a bit difficult to measure the differences between varieties that might be a little more subtle, and you might need a little bit more precision. So one possible solution is utilizing multi-spectral imaging to quantify this barley growth. So what is multi-spectral imaging? It's been demonstrated as an effective method in small grains to measure vegetation growth. And multi-spectral imaging is essentially using aerial imaging such as an unmanned aerial vehicle or drone, and having a camera that picks up different band wavelengths, and you can use those wavelengths to measure the growth rate of the barley. And to measure growth rate, you can't just measure at one point in the season, you have to do it repeatedly over the life cycle of the barley. So the goal is to first year characterize what point of the barley growth stage is the most heritable, and then use that as a way to figure out which lines might have higher vegetation growth and those might repeat the weeds. So this is kind of the flight aerial imaging kind of process in an overview. We do about 7 to 8 flights a year, about 20 to 70 days after planting. We did this for 2 field seasons and 2 trials per year, so a total of 4 trials. And then for each flight, you take calibration images before and after the flight. And in addition, it's good to have a ground truth evaluation. So we used a GreenSeeker NDVI to do that. And of course, you need to, when you perform imaging, you have to make sure you conduct the flights within 1 to 2 hours of solar noon, and try to make sure you have a either perfectly clear day or an evenly cloudy day, which isn't always a given in New York. So this just going over the kind of the overview of this processing of imaging collection. You get the images and use like different imaging software to stitch the images together, and that kind of puts them into this like kind of mega image, so to speak, that you can get a lot of information from. So you upload those stitch images, and then I've been using a program called ImageBreed out of the Breed-based system here at Cornell with a colleague of mine, Nick Morales, who's been really instrumental in helping me trying to get the information that I want from these images. And so what you do from that is you develop these plot polygons around the plots of your image, as you can see here, like each plot will have its own polygon around it. And then you can extract the pixel values or the bands, and then you select kind of out of that information what indices you're interested in. And so these model pixel values and indices are based on growing degree days relative to each year. So it's growing degree days kind of accounts for the rate at which barley grows throughout the season, as opposed to just simply days after planting. And so this is kind of what it looks like. I'm still working on different modeling approaches and kind of analyzing longitudinal data. Does involve a little bit more calculus and a lot of statistical methods. I'm also going to be a little more stringent, removing certain event dates where not statistically significant, possibly include weather information to explain some of these factors and the changes in the data, and expanding this use and been doing imaging this past year for a conventional winter multi-barley breeding program. And acknowledgements, USDA OREI project is what gets me through this. So I'm really appreciative of the funding for that to work on these projects. Aerial imaging with weed competitivity has been really exciting to learn about. Like, I've been really fortunate to learn how to do aerial imaging, but it's also been challenging because trying to figure out how to accurately assess weed competitivity for crops it's not an easy task. So any improvements that we can make even a little bit will hopefully move things forward. And yeah, so if anyone has any questions, should we wait till after?
Yeah, we're gonna wait till the end. So
Okay.
yeah, if you want to stop sharing, we can have Chris up next.
All right.
There you go.
Perfect. Hello, my name is Chris Massman. I'll be presenting some of my research with the Naked Barley Project, specifically my work studying threshability. First, we'll go through a bit of an outline of my talk. We'll start with an introduction of what threshability is, our objectives with this study, and the materials and methods used, some results, and finish up with the conclusions of what I have so far. So the first question is, what is threshability? Our definition is the propensity of grain to shed the hull after harvest and cleaning. Even in naked barley, there's some degree of hull attachment. And this trait is scored on a 0 to 4 basis, as seen in the figure to the right. Here, with 4 being no hulls attached and 0 being all hulls still attached. Threshability is important because if a naked grain fails to shed its hull, it must be dehulled like any other covered grain. And with a standard of 5 for food grade and 15% for feed grade, the margin for error is fairly low. Despite this trait's importance, not much is documented on its genetic and biochemical architecture. We do know that it can be related to grain size and shape, and that it is a highly heritable characteristic, but not much beyond that. Therefore, our objectives for this work were to first identify QTL or quantitative trait loci associated with threshability, to incorporate these QTL into a genomic prediction model, and decide if they can increase predictive ability. And then finally, test grain shape and weight characteristics as covariates in genomic prediction, and see if they increase predictive ability. First, a little bit about the germplasm used. There were 350 genotypes grown in 9 environments as a part of this work. Trials were grown in Wisconsin, New York, and Oregon over the 2018 to 2019 seasons. Modified augmented experimental design was used in all environments. This table here shows the environment proportion of genotypes with at least 2 replications, the number of genotypes in each environment, mean threshability, and broad sense heritability at each of the tested environments. Lines were genotyped, like Brigid mentioned, using the Illumina 50K SNP chip, and threshability was visually scored in each environment. Additionally, percentage of thin and plump grains, as well as grain test weight, were recorded for the Oregon environments only. I won't spend too long talking about my models today, but just so you have a general idea, mean performance of each genotype for each recorded trait was calculated by a 2-step mixed model. The first step accounts for spatial variability. The second estimates environmental and genotype by environment effects. The result is a single corrected value of threshability for each genotype that we will use in later steps. Genome-wide association study was used to identify QTL. Specifically, we used the Q plus K model for GWAS. This allows us to account for population structure and marker-based kinship. Brigid talked a little bit about the different groups that they identified. And basically, we are taking that into account when we are trying to identify quantitative trait loci. Genomic prediction was done through the GBLUP model, or genomic best linear unbiased prediction, using the significant marker associations, test weight, and plump thin scores as additional fixed effects. Basically, we're trying to improve our ability to genomically predict using multiple traits that we have phenotyped to predict the trait that we don't have phenotyped yet. For our Qtl discovery, there were 2 significant Qtl identified in the GWAS, one on chromosome 2H and one on chromosome 3H. They had effects on threshability of -0.18 and -0.29 respectively, and that is on a 0 to 4 scale. These associations are significant at a family-wise error rate of 0.05. In terms of our genomic prediction results, predictive ability was as high as 0.84, which means that there was a 0.84 correlation between the observed threshability and what we predicted with the model based solely off genotype. This is very high, and I was very happy to see that. Unfortunately, using our grain size and shape characteristics, we were not able to increase accuracy beyond that point. But even so, we can very reliably predict the threshability of a genotype based on its genetic makeup, at least according to these results. So in summary, we detected 2 significant QTL. Prediction accuracy was high, but unaffected by the use of additional covariates. Because of the presence of significant QTL, as well as a high predictive ability, either marker-assisted selection or genomic prediction merit further investigation as a tool breeders can use to improve threshability. Regardless, production of high threshability naked barley is an important step in increasing the utility of this crop. If you have any questions, please don't hesitate to email me, cgmassman@wisc.edu, and thank you.
Thanks, Chris. You can go ahead and unshare your screen, and I know you have to go early, so take off whenever you need to, and I can do my best to answer questions for you or direct folks to you. And with that, Cristiana, if you want to go ahead and share your screen.
Okay, so sorry about that. So I'm Cristiana. I am looking at characterizing embryo damage in naked barley, and like Brigid said, I am a master's student with her here at OSU. So what is embryo damage? Embryo damage is basically complete or partial damage to the embryo during harvest, seed cleaning, or malting. So here we can see 3 pictures of varying damage to these seeds. So here on the left, we have a fully intact kernel with the full embryo. In the middle, here we have a partially damaged or seeds with partially damaged embryos that will possibly not germinate. And then the final picture on the left is seeds without an embryo. So this embryo has fully basically popped out of the seed. And so we see this a lot in naked barley because of the lack of hull. So in covered barley, that the hull often will protect the embryo and the seed during harvest or seed cleaning. But because we don't have that, we see higher susceptibilities or higher susceptibility to this damage. So a result of this embryo damage is poor, uneven germination. So you can see here in the picture on the right is in the middle is one of our border rows that just had a very, very poor stand this year. And it's because these plants didn't germinate due to this embryo damage because of harvest. And so Karl talked about weed competitive ability, and embryo damage can definitely impact the plants, the barley's ability to compete with weeds when the stand is pretty low. And so we don't want to see this in producer's field. So I'm going to go into my experimental protocol. This is ongoing, so this is just currently what I'm doing. So I'm working with the spring diversity panel, which is 254 genotypes, as Brigid mentioned. And we collected individual heads from the field this summer. And we are threshing them at 2 different speeds, either 2700 RPMs or 3600 RPMs for either 2 or 5 seconds. And our control here is just threshing by hand. So once we've threshed those heads, we do a total seed count, and then count the number of seeds that have visible embryo damage. So like those pictures we saw earlier. And then we germinate all of the seeds using a Ragdoll method. And so if you're unfamiliar with this method, that's basically just laying out seeds in this germination paper, rolling it tightly, and then letting it soak in about 2 inches of water for 7 days. We then unroll that germ paper and then count the number of seeds that have fully germinated. And so that's, we're considering that to be both shoot and root growth from one seed, seeds that have not germinated at all, and seeds that have partially germinated. So that's either only shoot growth or only root growth. And so here you can see kind of what that looks like once we unroll these germ papers. So zooming into that top picture, this is 3 different genotypes, each threshed for 5 seconds at 3600 RPM. So this is our most intensive treatment for this experiment. And so here you can see in the top left, this genotype barely had any germination, and if it did, it was these sad looking shoots that partially germinated. And so there's that. Whereas if you look at these other 2 genotypes, we have still a significant amount of seed that didn't germinate, but we also are starting to see more partial germination as well as full germination. So this is based on the work that we have done with this, we are starting to see genotypic differences, which is a really good sign and is leading us to believe that we can start selecting for this trait. So along with the threshing and germination, we also are calculating the head shape and the seed shape. And so I used a digital microscope to basically calculate this seed size coefficient, which is just the length and width of each seed, and gives us a general idea of the differences between these genotypes. But moving forward, I'd also like to look at the embryo shape, the size of it, and the protrusion. So as you can see here, these 4 naked barley genotypes from the spring diversity panel are fairly different in their overall seed size and shape, as well as their embryos. So like I mentioned earlier, so we're looking at partial germination, and these could be seeds that looked fine, and we didn't see any like real damage to that embryo just by visual assessment, but are still partially germinating. And so we think this is caused by what we're calling micro damage, which is non-visible fractures to that embryo. So with this part of the project, we are going to select 12 representative genotypes from the spring diversity panel and take images of the embryos using a scanning electron microscope that we have here at OSU. And then from there, we can calculate the area and frequency of the micro damage to these embryos. And with that, I'd just like to say thanks to Barley World. This has been a really fun project. And here's all of our contact information, as well as our Instagram and Facebook. So thank you.
Great, thanks Christiana, thanks Karl, and thanks Chris who had to go. I think we're now gonna go ahead and open this up for questions. So we have about 20-25 minutes left. And so feel free to either use the chat, or I think you are all officially muted. I don't know if you can unmute yourself. It looks like yes. If you prefer to unmute yourself and ask a question, go ahead.
Question for you, Brigid.
Sure.
That fusarium is a huge problem for organic producers in Wisconsin. So I was very surprised by your results in terms of the survey. But is it so that naked barley also is more problematic with respect to fusarium? And what sources of resistance to fusarium are you utilizing for protecting our farmers in Wisconsin?
Yeah, great question. So I was also surprised by that. It looks like we actually have Brian Baker in the Zoom room, who was the one who conducted the survey and did the interviews. And we did get a very nice representation of the US in our respondents, but I was also surprised by the sort of lack of issues that people had with diseases. But we do have, so here in Oregon, fusarium isn't a huge deal yet. It hasn't quite moved this far west, but Karl, Chris, and we have another graduate student in University of Minnesota whose entire research project is actually looking at fusarium head blight in the diversity panel. And I was actually hoping to get him on here today, but he couldn't make it. But what he's looking at, so people have suggested that naked barley may actually show higher resistance to fusarium head blight because the hulls are threshed off. And so a lot of the fungal pressure is contained in the hulls and left in the field during harvest. And so the basis of his research is actually looking at the disease pressure as well as DON accumulation. So for those who don't know, DON is short for deoxynivalenol. It's a vomitoxin. And so in addition to fusarium head blight being a problem for yield reduction and quality reduction, it can also cause this vomitoxin, which can be toxic to humans and animals. And so he's looking at accumulation both in the seed and in the hull to see if there are differences. Yeah, so stay tuned for those results. And then, Brian, if you want to chime in really quick on the survey, but then Karl also, if you want to, we do have fusarium testing nurseries that the trials are going into, in both in Cornell and Minnesota, and then I think in Wisconsin, they're just recording natural infection. But yeah, Brian, if you want to speak really quickly to the survey.
Yeah, a couple of quick points. Thanks, Walter, for asking. And I was as surprised as anybody. I mean, I think we all were surprised. But couple of things. Another thing that I also found surprising was that most organic producers of barley are growing for feed, not for brewing or malting, not for culinary use. In the interview part, several of the producers I talked to said, oh, well, yeah, we've got diseases, but they're not really costing us much in terms of yield. They're out there. We don't really know what they are. And some speculated on what they might be. There was definitely a regional difference. And in the Northeast and for many years in the Midwest, the North Central region, diseases are much more of a problem than they are in the West. And but yeah, it's seen as something that's not a limiting factor on a lot of the farms. That would change if people went to higher value uses and barley was more important in their rotation.
Great, thanks, Brian. That's a great insight. Yeah, Karl, did you want to speak to Fusarium? And then we have a question for you in the chat.
Yes. So Fusarium, we're not testing on the diversity panel, but most of our lines that are in regional testing or our naked barley variety trials, we test an inoculated system here at Cornell. And so we do, it's an inoculated system that accounts for heading date, and we have a mist system too. So we do that for both winter and spring barley and wheat and occasionally other grains as well. So we tend to find, we tend to get the most consistent data and conditions for the winter barley compared to the springs. But there's definitely a variation in susceptibility and resistance. Regarding what?
I was just going to read the question, Karl.
Oh, yes. Okay.
So this is a question to Karl. How do you normalize the soil background if there is any discrepancy in moisture content between plots? Do you use the OSABI calculation instead of NDVI or other methods?
Yeah, so I did look at the soil. I did not see a bunch of variation in the soil. I did look at the SAVI because that's one of the indices we calculated, and I didn't see a huge discrepancy between the plots, but the OS, that's another one that I need to look at because I just quickly looked up because I know of soil adjusted vegetation index. So that's what SAVI is. And this the OSVAI is something I need to look into a bit more. But yeah, I can take that one to account and see if there's a difference. But overall, I didn't. We fly at about 25 meters. That's the other important part. About 25 meters above the plots, but I did not see any significant variation between them for SAVI or for the discrepancies of soil background, so to speak.
Great. Any other questions?
Brigid, this is Cooper. I have a question or a comment.
Sure.
So I'm coming from the perspective of a producer in the arid West who farms tens to hundreds of acres of small grains every year. And I just want to sort of tease out these comments on disease a little more. And I would just offer that most Western producers are growing anywhere from, 3 to 8 other crops in rotation. And we really are often growing barley to feed cows, not feed humans. And so the toxins issue from disease is really an afterthought for the most part. There are only a few people who are concerned about that unless they're perhaps growing barley for food and their handler or their customer is actually requiring, testing for toxins. And it, at the risk of sounding uneducated or, somewhat disparaging to myself and my fellow farmers, those sorts of specific disease things are not on our radar unless we're growing for malting and we have our malt customers, agronomists on speed dial. And we'll call that expert and say, look, I got an issue. I don't know what's going on. And that person will come out and take a look and guide us. But the producer's knowledge is probably far less than anyone really expects, to be honest.
Yeah, that was something that I think I picked up on a little bit. I've mostly worked with barley growers who are growing for food or malt. And so, yeah, I think depending on your end use, it makes a big difference in the quality that's needed. And so if you don't need the quality, then you're not going to spend a lot of time trying to control the disease necessarily. And I do think more education is needed because I know, for example, for covered smut, that didn't even show up on that survey, and it was a category that we put in, but no one seemed to have an issue with it. And I think some of the problems there is that people don't know the difference between covered and loose smut, and really why should they, except that it does make a difference in how it infects the grain, and also whether you're going to end up with smut balls in your harvested grain. And we got some organic malting barley to use for one of our projects, and yeah, it came full of smut balls, and I don't even know if the growers really knew what that was. It's just interesting to me, sort of the disconnect in what breeders think of as important and what growers do. And we've identified smut as this huge issue, and maybe the growers don't really care, or maybe they don't know that they should care. And so that's sort of what we're trying to parse out. But ideally we would breed for resistance, and then they don't need to care because it wouldn't be an issue. So,
Well, to add to your priority list, although it's a little late in the process,
no
we are
problem.
we are seeing increased use of small grains, particularly winter hardy small grains, as cover crops in organic production systems. And so especially in our approach and in our farm itself, finding winter hardy cereals that can survive in the Intermountain West and bare ground, uncovered through the winter down to 0 degrees is really valuable. We only have a very limited toolbox. And in terms of a soil health perspective and the big production system, the big picture, we just need to beef up that toolbox. So to the extent your work and the team's work collectively produces more winter hardiness in new varieties, that would be much appreciated.
That's definitely great to hear. We don't, our collaborators on this project are sort of in the northern part of the country, and we would love to get more researchers or growers who are willing to do trials in other areas, because we don't have anyone in the Southwest or the Intermountain West. And so we're a little limited in testing.
Yeah, so for the winter grains, some of the winter grains have actually been grown in Madison for a year, and they were successful, but most of the winter or fall variety trials are in Oregon and New York, and more specifically in New York, we do get differential survival and winter kill, but that's not very helpful for out in the West, because in New York we get snow cover and whatnot. Kevin Smith is in Minnesota is trying on conventional barley for winter hardiness in the germplasm, but again, it's a little more difficult because when you have that bare ground and frigid temperatures as it gets out here, that can be difficult.
Yeah, we have the Madison and Minnesota and St. Paul locations have grown the fall regional trial, and the first year got complete winter kill, I think, even with some snow cover. But the last couple of years, we have been able to get a bit of differential out of them as well. So we should have a bit of an idea, at least in the germplasm that we currently have, what is more winter hardy than others. But again, I'm sure you're familiar that wheat is overall more winter hardy than barley is. But there are certainly things that we can work on to improve that. And winter hardiness is a very important part of the overall Oregon State University barley breeding program. And so we're definitely working on that, which is a little ironic given that our location never sees winter kill. But we do a lot of regional trialing and national trialing. So yeah, and Brian added a comment that winter hardiness was included in the survey. It was important in northern states. Drought tolerance was a slightly higher priority in the in the West, and we actually, with our Davis location, they only joined the project in 2020, but we're hoping they have certainly been seeing droughts during the growing season. And so we're hoping we get some good drought tolerance data from them. Yeah.
Well, thank you. Thank you for these updates. It's very encouraging.
Yeah. And, you know, we, especially with our regional trials, we're always happy to send those out to additional collaborators beyond just those who are in the grant. I know this year I sent some lines to Kansas and somewhere else that I'm blanking on right now, but if you work with a research station that does trialing and they're interested, we would love to send lines for them to trial.
Well, thank you. I'll keep that in mind. We do try to engage with our University of Idaho extension groups pretty actively, so we'll add that to the list.
Super. Yeah, I know University of Idaho, I guess we mostly work with the USDA folks in Idaho, but they don't have organic trialing sites that we found, so we haven't been able to involve them in this project specifically, but we can also send to conventional locations if that's all that there is. Thank you. Yeah.
Got another. Be in touch. Two questions for Karl. Karl, I assume that look at those barley plants too and have an assessment for which of them is actually very vigorous or score them in some way. But so as to what are the most competitive barley, where do they come from? And the second question is underseeding. A lot of organic farmers actually underseed their small grains. That's part of their place in the rotation. And that does help with weed control, but it might also be a place where you could evaluate, how competitive the barley is. So I'm just those are two questions I've got for you.
Could you repeat the first question? Sorry, I'm paying attention to the second.
Where's the most competitive barley from? And the second question is, have you considered underseeding and evaluating the barleys in relationship to underseeded legumes, for example?
Okay, so I'll have to follow up with the first question on that because I did have the answer to this and I blanked on that one. Following up on the second question, that is, yeah, so this is like the part of a collaborative where we'd have to work with agronomy and whatnot, because we have in our program at Cornell with organics have done some trials such as that with wheat, with red clover seeding and such as that. But yeah, that might be a good option to do, is do an underseeding, so then you can have a little more of a consistent or lack of variation in the field. But yeah, that is a good idea.
Can I just add something in there real quick, Karl? I believe actually the folks in Wisconsin, so Chris and his group are underseeding their trials with a medium red clover. And we actually tried that here in Corvallis last winter and none of the clover germinated. We don't really have as many issues in the winter plots with weeds as we do in the spring. But intercropping and underseeding is definitely something that I'm interested in. But as Karl mentioned, we don't really have an agronomist per se on the project. We're all breeders. And so we are looking to put in a third round of this project likely for 2023, and that is management is a component that I would like to work into that, and that specifically both barley for cover crop and interseeding is something I'd like to work on. And then Karl, did you want, I have some thoughts on that first question also. If you...
Yeah, so just to follow up, I will say that if you're in an environment where you can grow winter organic barley, I'd suggest the winters over the springs because of the fact you can plant it in the fall, and that serves as a cover crop like function, and so to speak. And yeah, if you don't plant your spring grains, like in New York, for example, the weeds become a huge issue. But yeah.
Yeah, so in addition to the weed competitivity aerial work Karl has done on the regional trial, all participants do a weed competitivity assessment, which involves, we do seedling counts early on, and then we do usually an early vigor and early plant height rating. And then we do weed estimates using a quadrat. So we're not, we're estimating barley percentage, open ground percentage, and weed percentage throughout the growing season. And I can't say that we've noticed that there's like a specific breeding program that the most vigorous lines are coming out of, but I can say that there are certain types of plant architecture that do seem to confer greater weed competitive ability than others. So the sort of combined prostrate and erect growth habit, especially over the winter, seems to be the best. So ground cover as well as shading. We see the extremely prostrate ones. Sometimes the weeds will just grow through them and then they don't have any shading. And the ones that are very erect, there's more open ground, but the ones that sort of grow up and out at the same time are really the most effective, as well as, so that would be getting sort of at leaf angle, leaf width, the width of the leaves also makes a big difference. And so we have identified lines that do have better weed competitive ability in the regional trials. And unfortunately, we've been just a bit slow in sort of compiling all these data, but ultimately our goal is to get sort of basic summaries from all the trials onto eOrganic, where that can be available for anyone who's interested. And yeah, so I see.
Great, so much.
Yeah, no problem. I see one more question in chat and then, or a comment perhaps. Okay, Yadi Wang, am I getting that pronunciation correct? Is a farm manager at Oatman Flats Ranch, regenerative organic certified farm growing small grain in Arizona, several locations. Oh yeah, we would love to have, we'd love to send you seed to trial. Did you get my email address off that presentation? I can put that in. Oh, this was actually just to me, sorry. In any case, does anyone have any other questions? No, well, I think we're about out of time regardless, but thank you all for coming to this session and having interesting questions and conversations. Please feel free to reach out. And you know, we're 4.5 years into this project, and really we're just starting to get like the first round of publications and results out. Breeding, and especially breeding sort of a weird crop is, you know, we're sort of starting, I don't want to say starting from scratch here because there are a lot of people who've done great work in this for a long time, but there hasn't been as much work done as in other crops. So we are starting from perhaps,
Where to? I don't know.
yeah. So anyways, yeah, thanks all for attending. I just want to let you know that there are some sessions coming up starting at 2. So we have vegetatively propagated crops and a meetup for organic seed course alumni, as well as a happy hour at 4. And then the conference starts up again tomorrow at 9 a.m. Please take some time to fill out conference evaluations. And yeah, feel free to reach out. We do have an eOrganic website as well as some social media if you would like to keep up with the research that is currently ongoing and the results that will be coming out soon. So with that, I release you out to the rest of your day.
Thanks a lot.
Thank you.